Good evening, everyone. I'm Dayong Jin from Faculty of Science. Today, I'm here to show you a couple of examples how we are going to use light to diagnose and treat the disease. I'm an ARC Future Fellow and also the director for the new Institute for Biomedical Materials and Devices, and also the director of ARC Research Hub for integrated devices for end-user analysis at new level, ideal. As you can see, this hub is a highly focused on industry research and user-driven research. 2 weeks ago, we had a wonderful hub official launch at UTS. Everybody very happy. The key focus for this hub research is to develop a portable, easy-to-use devices for disease diagnostics. I'm holding this device. You could be very familiar. You can spend $10, buy 3 of them for pregnancy test. The result could be very easy to read, and the device itself it can be quite reliable. So the question is, can we develop some similar technology which is portable, but we can detect more complicated disease, like a cancer? So the answer is yes, we can do this by collaborative research. So this is an example of our hub to working collaboratively with the University of South Australia and also 4 Australian industry partners. We try to put together the latest development of technology like biotechnology, nanotechnology, photonics technology, which is the light technology I'm going to focus today, and microfluidics, communication technology, integrate all of this technology together into a small devices. If you can remember, back to 10 years ago, everybody want to integrate music player, mobile phone, internet, GPS together into a small handheld device. That was a dream, 10 years or 20 years ago. And nowadays, you can see the telecommunication technology already enable a mobile phone version device for you to do a lot of things, by one devices. So for the biotechnology detection devices, and this is our design, you can have a lot of nano materials, which is porous. Also have a photonics material, which is give you a very bright light. You can directly see the disease. And also you have a analytical chemistry technique developed for the last couple of decades, and then you can bring them smaller, okay? So to build a portable, truly portable devices for a single test, for a very simple test. The detection, in this space, it goes much beyond than detecting cancer, You can also use this detection technology to detecting the circulating tumor cells and to understand the stage of the cancer patients. Also, you have, this is a similar problem really to detect the pathogens and infections in our water, in our food. But the requirement can be very high. If we have more than 10 cryptosporidium pathogen in 10 liter large volume of water, and when you drink it, you can get sick. So we define this detection and sensing technology as a single problem. So we call this as a needle in the haystack single problem. Once you can detect single, you know everything. Okay. So as a physicist by training, many years ago, we start to summarize. The key challenge. And what's the typical challenge in this field? So we start to think, as a from physics point of view, they are the sensitivities, which means your detection has to be sensitive enough, and the speed has to be fast enough. High throughput means you can simultaneously detecting multiple analyses in the same time. And resolution, we spend a lot of time in the recent years to challenge, what's the highest resolution? We can see a single cell. We can see a subcellular structure. We can see even a single molecule. At this level, you could have a much better understanding about the fundamental of life. When I show this picture to many people, you have an impression. What are they? What are they? Australia? Actually, they are not Australia. They are tiny, tiny little single cell. They are single platelet cell under a fluorescent microscope. Using this traditional fluorescent microscope, the best you can do is you can see something similar like this, right? but use a new technology we developed in the last couple of years. We call super-resolution technology, super-resolution microscopy technology. You can see lots of more details into the subcellular level and into the organelle, single organelle level inside the cell. So by providing this new map, I call map, you could have a much better understanding of the fundamental of biology. For example, many years ago, you probably remember, when we drive, we rely on a printed map, which give you quite limited information, okay? Very fixed and very limited information. But a couple of years ago, Google did something really clever. So they combined the satellite image and sending cars with cameras, with sensors around every street, and you collecting all the images from different streets, you correlate these images into the satellite image, then you can build a street view. In the biology, this is our dream. We want to apply different technology, different imaging technology, different nanosensor technology, and build the inside, the street view version of inside single cell. Okay. Once we know that, you could have a much better understanding the fundamental of diseases, and you could monitor and even predict the disease, or monitor the metabolism status of a healthy human being as well. So before we're talking about how we use light to realize all of these dreams, I have to show you, what's the fundamental of light, and what's light? We all know God created light before the God creating us. So what is light? Light can be conceived as two different physical models. So one model is a quantum picture. The quantum picture of light, talking about the light is actually have many particles. We call it photons. When we talk about electricity, we know the fundamental element of electricity is electrons. The same goes to light, and the fundamental of light is electrons, is photons. Okay, so this little movie shows you the latest outcome. From a scientist. They use a super fast camera. They take images every 20 picoseconds, and you can record how a batch of photons they've been bounced back by a mirror. Okay, the photon like a tennis ball. Okay, so when you hit a tennis ball on the wall, the ball will be reflected back. Okay, so this is a photon meet the mirror in the femtosecond pulses, ultra short pulse, still with like billions of photons, quite a lot. And then once the photons hitting on the mirror, you got reflected. So this is called reflection. Okay. The other model we call it they are wave model. So as we know the sound, each different sound, they are the vibration of they have different vibration. The different frequency give you different tone. But this one is light. When you have a white light in front of passing through a prism, the prism will separate the light into different color. And the different color representing different frequency, the light wave frequency. Then you have different, you can recognize different color. In biological world, biotechnology, we use a color quite a lot. Use color to differentiate different biological substance. Also, when light have a interaction with matters, like when you get a light hitting on a material, and in particular this material, we all know jellyfish, right? So when you're shining a UV or blue light into a jellyfish, the jellyfish will emit beautiful green. It's very colorful, beautiful green. What are they? So they contain some material called protein. So this special protein, they will receive the light and slow down the frequency of the light. Okay? So by slow down the frequency of the light, you can change the color from blue or UV blue to green. And by this way, you can separate your excitation light and the emission light. And you can use this special material as a probe to label your target, like a cancer cell or even subcellular structure. Then you shining a light by blue, then you check whether this material got green and where you got the green. And by this way, you build a fluorescent microscope. So this is really the fundamental structure of a fluorescent microscope. They use a mirror. So this mirror, they have quite a special property. So when you have blue light hitting on the mirror will reflect the light. But when you have a red light hitting on the mirror will transmit a red light. So this special mirror is the most fundamental element in the fluorescent microscope these days. I want to do a simple experiment. I take a mirror out of my microscope in the lab. This is a very simple mirror, but when you have a red laser, you can see this is a red laser, and you probably can see from the other end, so this is a red laser, right? So that means they are transmission. But once you have a green laser, the light, the mirror will reflect the green, right? So you wouldn't see the green passing through. But from the other side, you see this mirror will perfectly reflect all the green. So this is the principle for all the fluorescent microscope, by using this mirror and to separate your excitation light and emission light. When we're talking about fluorescent microscope, there are two very important recent Nobel Prize projects related to fluorescent imaging. One is a 2008 Nobel Prize in chemistry to award 3 scientists who initially discovered there exists of fluorescent protein in jellyfish. And they can separate this protein out of jellyfish, and you can use this protein to label the single cell. And you can see, once you separate this different color of a protein, and you can specifically label to different compartment of a single cell. The red one is a cell, membrane proteins, and the green one is the microtubule structure inside the cell, and the blue one is a cell nucleus, which is the headquarters of a single cell. And also, you can use different color to label bacteria, to understand, to diagnose the infections. The other very recent Nobel Prize in 2014 award to 3 scientists who independently developed different technology to improve the resolution of a microscope. So this is a traditional resolution, which is a traditional, high-end fluorescent microscope can do to see a single cell. But this one is a super resolution. You can see the super resolution provides much better and more detail for biologists and medical doctors to understand the basics of the disease and the single cell. In this field, our researchers at University of Technology Sydney has significantly contributed to the development of super resolution. This paper we published last year and talking about developing a new super resolution technology to deliver the highest resolution in the world at the moment is 19 nanometer resolution. So what shows in the picture? So this is a single cell inside. If we think about single cell like this big, and we think about the nuclear like this big, this one is on the surface of a nuclear. Each single cell nuclear, they are like the headquarters. They're sending signals, to the cell and ask them what to do. Okay, so by chemistry, by biological molecules. So what we're trying to image in this experiment is to see, how the nuclear they send the molecules and chemistry outside, from inside to outside, through a structure called nuclear pore complex. So this is a enlarged picture from here, and this is a further enlarged picture. And you can see this is a ring structure, like an O-ring structure of a nuclear pore complex. And now the optical resolution allowed you to see this tiny little small hole on the nuclear for you to understand, the live cell communication and structure information.
A tiny mirror could make a big difference for scientists trying to understand what's happening inside living cells. Cells are three-dimensional, and that's a problem for scientists trying to see pores and other tiny structures. Existing microscopes provide high resolution in the X and Y axis, but not the Z axis, which is what scientists see when they look directly into the cell, perpendicular to the glass slide on which cells are usually examined. But by growing cells on tiny mirrors and imaging them using super-resolution microscopy, a team of scientists from Peking University, the University of Technology Sydney, and Georgia Tech are addressing this challenge. Their new technique creates interference patterns as light waves pass through a cell on their way to the mirror and then back again after being reflected. The interference patterns provide, at a single plane within the cell, significantly improved resolution in the z-axis.
All right, so you can see this is a simple example to show you the complicated, more powerful technology doesn't mean you have to use complicated technology. The technology itself is very simple. You can use a simple, a mirror, rather than grow your cell on the glass substrate. And this new technology provides a lot of more scope and a lot more information for the biologists to understand the basics of life science. And also is a typical example for international collaboration. So in this case, we work with scientists from China, from US, United States, to get this project delivered a couple of years ago. All right, I'll show you another example, how we use the other different dimension to separate the target cell from the background samples. So we call time result microscopy technique. This picture is the microscope I built right after my PhD study many years ago. At that time, we have quite limited resources to build, more expensive microscope. So what I can do is find the old generation microscope, and you can see this is an old generation microscope. I chop the head off, and I add a decorative mirror, the mirror I showed you just now, and then you can build a UV LED as excitation, and then you're collecting the red emission from the target signal. The more special, powerful, uniqueness from this microscope is this one allowed you to see the target without background. How we do that? It's pretty much like the scenario when everybody is sitting at home watching TV, and suddenly the house getting powered off. Okay, so then you see everything else, the lights, everything else went to darkness immediately, but the screen of your TV will last a little bit longer. Okay, so this is a special material which has a slightly longer fluorescent lifetime. So in biology, we use a material quite similar to this material. We can use this material to label, in this case, a Giardia, single Giardia cell with a long fluorescent lifetime. Then by using a pulsed, the flashlight to excite every sample, and during the flash time, you have a optical chopper, means you have the chopper blade, the fast rotating. But during the flash time, the chopper blade will block the access for detection. But after the excitation light, the chopper blade move away, and then the short fluorescent material, they disappear immediately, and then the long lifetime material will stay in the picture. So this simple technology, again, allowed you to directly see the single cell without any background. Okay? So you can further apply this technique to apply multiple color, the lifetime and the color. And you can simultaneously detecting Giardia, and you can also count how many cryptosporidium from green label. So by this way, you have simultaneously high throughput detection, multiple analyze in one image. And we further extend the technology to detecting cancer. So this image shows you a prostate cancer cell. Okay, so we label this red fluorescent material with on the red cancer cell, and we image this cancer cell out of urine, because urine, we know you have a lot of background information, and it's very hard to directly find them. Find the needle in the haystack, it's very hard to find them. But by this way, because you remove all the background information, and you can clearly find where are the cancer and how many are they. Okay, so by doing this test, you probably can, we are working really, collaboratively with this Sydney-based company called Mnemonic International, and they developed the protein technology for you to directly, specifically find the cancer cell. And we're trying to put together this research effort to develop a direct technology for you to find how many cancer cells from the first urine in the morning, and then to diagnose, the risk of prostate cancer. And the current method for prostate cancer detection is PSA test. 3 out of 5 positive PSA PSA positive patients can be false positive. Okay, so then you scare a lot of people, and most seriously, this PSA test can miss a lot of patients who already got cancer, but the PSA result shows negative. So this is a quite big problem. But by developing this sort of technology, you can directly count how many prostate cancer in the first urine in the morning, and then significantly improve the detection accuracy. And also, by the way, this is a non-invasive detection method. It's a lot of more acceptable than the biopsy testing. So this, we believe, going to be the future of prostate cancer detection. The other challenge is when the disease start to come, they only express a very little amount of disease biomarker. So this is a typical image we take in the electron microscope, which has a much higher resolution, but of course, $1 million machine. And then you can see on the surface of, in this case, is a leukemia CD34 cancer cell. And you can see on the surface of the single cell, you have quite a limited number of biomarkers. This is particularly true when the disease just start to come. And the challenge is, can you quantify the number of biomarkers per single cell, which you could require much higher resolution and you require much higher sensitivity. So in the recent year, we developed this technology called SuperDot technology, which is very exciting. My team currently, majority of my team are focusing on this technology now. You can see we start to use a nanotechnology. We can develop a lot of well-controlled single nanoparticles at different size. But the uniqueness for this particle is they can combine multiple photons. So in electricity, it's very hard to combine 2 electrons into 1 electron. But in photonics, you can combine multiple photons, 1, 2, 3, 4, even 5 photons together to emit single photon. The advantage for this would be you can use a infrared light, which is a much lower energy, but they can penetrate much deeper through the tissue. And then you can combine the multiple infrared light and convert them into visible ones. And you can see the movie just now. It records how you make these nanoparticles, and you shine it by infrared light, and you can see beautiful bright green and blue fluorescent. So this will bypass a lot of problem in biology, particularly for this deep tissue imaging, because we know the light doesn't penetrate deep through the tissue. I got another experiment I want to show you, very simple. So this is a green light, right? So if I'm shining the green light towards my finger, and from the other side, I don't think anybody can pick up any green light. But if I have a red light, which is a less powerful laser, but you probably can pick up the red signal from the other side. Can you? So that means the red light, they penetrate much deeper. What about infrared light? The light near this infrared window, we call it optical transparency window. You can see, well, the laser one, I use around this wavelength range. But once you further move the wavelength longer into near infrared window, the tissue, the tissue will be, the light will transmit much deeper into the tissue. So this will allow you to use a light-based technology to directly see the tumor cell. So this is a new result we received, we collected a couple months ago with, again, a collaboration with Fudan University in China. You can see this is a pool mouse. We inject this material into tumor, all right? So by shining a near-infrared light, you can directly see, actually, where's the tumor. So by optical, optical is much simpler and easier. So this will allow the doctors to clearly see the boundary between tumor cell and the healthy cell, and you know exactly where to cut, okay? So this is an example, and also the latest research result. So far, we only talk about how to use light for diagnose your disease and detecting single cell and disease biomarkers. And you can also use light to trigger the drug release and for the therapy, for the treatment. So this is some latest the review paper by our collaborator in China, they put together a nice review and quite long article. But I really like this image because you can see by developing this material, which is a multi-shell and porous material with different components, the goal, and by the way, they are tiny. You see, this is a scale by 50 nanometer, and like more than a millions smaller than the diameter of our human hair. So very, very small one. So you can build multiple functions into one material. And this new material will allow you to carry the drugs, also allow you to do MRI imaging and optical imaging. When you put the drug, when you throw in the drug, each particle can have a tiny, tiny amount of drug. So much, much below the safety threshold. And this nanoparticle can recognize the cancer tumor cell, and you can use this infrared light to trigger this nanoparticle and let them to release the drug when you see they are already in the tumor region. So this guarantee a much safe operation of chemotherapy and radiotherapy. So this is a very promising technology for our future. In the same time, we try to study the fundamental science like chemistry and material science to see how possible we can control each single nanoparticle. And how possible we can start to build multiple functionality into one. So this is the case to show you, how we play in the lab, by growing different shape, different functionality into the nanoparticle, and different, structure of this nanoparticle. By doing that, you could develop a nanoscale sensor, exactly like, how the Google sending different cars to drive on each street to take the pictures from and build a street view. And if you have a different set of nanoscale sensor, you could start to have multiple sensor built into a single particle, and then you can drive this different particle to different subcellular compartment, and you start to collecting the localized temperature, pH value, the traffic condition intracellularly. So by this way, I think this is a very ambitious goal, but I think it's highly possible for us to achieve this goal. All right. By the end of my talk, I still want to emphasize the importance of collaboration. So the future technology really belongs to people who can build international, interdisciplinary, and industry collaboration and start to tailor the technology and the science and to integrate different technology together. All right, finally, I would like to thank the funding agent, which is the Australian Research Council, through different funding schemes, and also my team of students and postdocs who has working really hard and drive this technology behind. Thank you so much.